that. But where we are now is it's well established checkpoint blockade can give you durable cures in a fraction of patients, and so we need to make that better. Well, how do you make it better? Well, the best thing to do is, like I showed you with ipi/nivo, is to combine things. But if I can get off my soapbox for a minute, one of the things that I find very distressing now is that there's something approaching 2,000 clinical trials of PD-1 antibodies plus something else. Most of the time, that something else is purely empirical. There's no ration behind it, other than maybe a company owns 2 drugs. Oh, let's see if they work together, And then there's a clinical trial done. If they don't work, they throw it out, which to me is crazy and ought not to be allowed. If you're going to do that, you might as well do a biopsy and see what happened in the tumor and learn something. I mean, I've spent a lot of years down the hall from Lloyd Old, many of you may know of, and Lloyd's, one of his mantras is, if you don't learn something from every patient, you're an idiot, and so, and so what Pam Sharma and I do at Anderson, we've got a big lab, and we get involved, and we're involved in 100 trials right now where we just try to get specimens from 20 or so, matched pre-post on therapy with blood, and figure out what's going on, because you can learn as much from a failure as you can, from a success. And so to throw things out because they don't work in one combination is a real mistake. But, and I'll come back to this later, we really do need to know the cellular and molecular mechanisms of things we're putting together. Because of this next thing, we need to know the impact of therapeutic agents on the immune system. So people, throw epigenetic drugs at cancer, lung cancer, for example, and we know they could work in part by, killing some tumor cells maybe, but also they make them upregulate MHC antigens and upregulate other antigens that they express, and then also re-express cancer testis antigens. But nobody's ever looked to see what those drugs do to the T cells, because we know there are epigenetic changes in T cells as they're developing too. And so we need to look at both sides of that. The same for the small molecules, the TKIs. Most of those drugs have never even encountered an immune system in their entire development. And now people are throwing them out, and they don't have any idea if they even target that pathway in cancer or not. But anyway, we need to learn that so we can combine the best standard of care that makes sense, find some new molecules, checkpoints and others that we could use. And finally, it'd be great if we had predictive prognostic or pharmacodynamic biomarkers. What we're pretty good at now is patients that are on therapy. We can tell very quickly whether they're going to respond or not, but we cannot predict with any certainty at baseline, who's going to respond. So as I said, I want to talk about now the differences between these 2 drugs, because there's been a tendency, because of one thing, and that is that PD-1 it has exactly the same, well, not exactly, anyway, pretty much the same range of adverse events as does anti-CTLA-4, but lower frequencies, and so it's easier to use. And so that's caused a lot of people to say, well, let's just use the one. It's also known, however, through the work of Tony Rebus and others, that in about 2 years, there's a 15% or so relapse of patients that have responded to lung cancer and melanoma, and so there's this fall off, whether that affects the its response rate is higher than IPI, so whether that affects long term, we don't know, but that is a fact, whereas recurrence with CTLA-4 is pretty rare. Anyway, so very quickly, so I told you CTLA-4 is hardwired. It plays a role in every immune response in your body in stopping the T cells. PD-1's induced resistance. CTLA-4, there's some controversy, but seems to target CD28 at the time of co-stimulation in early differentiating cells before they're functionally mature, whereas PD-1 targets the TCR pathway on terminally differentiated effector cells. So that's a big difference right there, for reasons that I'll show you later. CTLA-4 has been shown, since it works at priming, to recruit new clones of T cells that have not been involved in a response, whereas PD-1 just gives you more of the ones that you've got. It allows them to keep proliferating when normally they would have been turned off. But it has not been shown to recruit new T cells. CTLA-4 almost didn't get approved because the responses are slow. So you have to, we had to switch the endpoint of the trial to overall survival instead of progression-free survival. Anyway, PD-1 is usually pretty fast. Again, it works on terminally differentiated T cells, so you don't have to generate the army after you've gotten it. So I'll show you CTLA-4 primarily affects CD4s, although also some CD8s. PD-1 affects only CD8 T cells. It has no effect on CD4s at all. Again, data that I don't have time to show you, but we've shown in prostate cancer and in kidney cancer at least that are cold in terms of infiltration, CTLA-4, but not PD-1, can drive T cells into them. Get other problems because CTLA-4 also induces PD-1 on T cells, so, but you can at least get the T cells into a tumor where they weren't before. I talked about adverse events. And then there's the dosing, which there's various depending on the drug, but usually CTLA-4 antibodies are given up to 4 times at 3-week intervals, and that's it. 3 months and it's stopped. And so people are still alive 10 years later, so that's sufficient, 4 doses, 3. Whereas PD-1 antibodies, depending again on the manufacturer, are given as often as every 2 weeks for as long as 2 years. And so very different dosing. And there is a reason for that. First, I thought it's just because, PD-1's safe enough where they could give it forever, and so you give it forever. But actually, there is a reason. Anyway, so the rest of this talk, I'm going to show you some data that I'm pretty excited about. I don't have time to show you all of it, but we wanted to go in a very, unbiased way and just ask, what are the T cells that are modulated by these 2 different drugs, and which are the ones that play an important role in tumor rejection? And so we did this using the technique high parameter flow cytometry, or CyTOF, where we've got up to 43 parameters we could look at per cell using antibodies tagged to heavy metal ions and other things. I won't go through the details, but basically you can measure that many colors. So what we've done in the study I'm going to show you is we've got T cell markers, enough other kinds of lymphocytes to get rid of them, focusing just on T cells. And we've got the service markers of differentiation, transcription factors, and everything that allow us to tell the different subsets of T cells. Also, we've got in the panel things that will tell us which cells are proliferating, which are not, which cells may be dying, which are not, all that sort of stuff. So we do the analysis. We also keep track of individual mice that we're looking at. And in every mouse, then we correlate the number of the different kinds of cells that we see with the tumor size. So we can say these tumors, not only these T cells not only went up, but the size of the tumor went down, because of it. Or it didn't change, because there are some changes that don't matter. Any event, so this is the scheme. I'm just going to show you one tumor model, but we've done this in a couple. This is a pretty immunogenic tumor. We've done it in a poorly immunogenic tumor with a vaccine, and we've looked at enough patients to know that we can't do it obviously the way in patients that we do in mice, but we've looked at enough where we see essentially the same things in terms of the cells that are involved. So this is called a phenogram, and so each of these is a cluster that we found was interesting. And it's not, I don't expect you to get much from this side except that each of the markers is here vertically, and each of those is a cluster. And so by looking up and summing what's in the vertical column, you can get an idea of everything that cell, pretty much everything that cell is doing. Of course, doing it here will give you a headache, as I learned when this postdoc first started bringing this data to me in this form, saying, pointing at a square and saying, look at that, right. Anyway, so here we've condensed it to the cells that are relevant in the main populations. So in Tregs, you can see here, the blue is an isotype control, red is anti-CTLA-4, green is PD-1. And so what you can see here is there's 2 types of Tregs. They both have CD4 and FOXP3. The one on the left has a molecule called KLRG1, whatever that has to do with anything, but the other one does not. In any event, both of those are associated with bigger tumor size. You might have expect. These are suppress the other T cells. And you can see that CTLA-4 for sure depletes. And actually we've shown this in other publications, Tregs. Should point out that this appears to be an artifact of the mouse antibody. The ipilimumab does not do this. We've not been able to detect this in any human patient that's been treated with ipilimumab. So just forget that for a moment. Let's concentrate on effector cells. So next are the CD4 effector cells. There's two here. One of them is what we call a Th1-like ICOS positive cell, for reasons that'll become obvious later. But there's plenty of Th1s. Those are the cells that make gamma interferon and TNF. But usually they don't have ICOS on them. These do. They're, as you can see, induced very high with anti-CTLA-4, not at all with PD-1. There's another population of cells which look like naive CD4s, or maybe just be bystanders that aren't affected by anything, and they don't matter. But anyway, these ICOS positive, TBET positive Th1s are associated very strongly with smaller tumor size. So these are the CD4 cells that are killing tumor. Go to NKT, just get them out of the way. Nothing's going on over there. But anyway, in the CD8s, what you can see, the CTLA-4 also expands these. Now, these are CD8 cells that are effector cells. They're terminally differentiated cells that have TBET, so they're killer cells. They have a transcription factor called the OMEs and KLRG1, which just says they're mature, they're fully functional. That's expanded by CTLA-4, and as you can see by PD-1, those are definitely associated with tumor shrinkage. The only other ones that are really important are this one here to the left, this cluster, which is labeled PD-1 high, TIM3 high, also LAG3 high. These are cells that are exhausted. They've got this triplicate of three very strong inhibitory receptors on their surface. Those guys are out of it. They will not divide unless you give CTLA-4, unless you give PD-1, and they divide. As you can see here, they increase in number. And also we've got markers in there, and we know that they're proliferating, since we gave the antibody. They are not increased by CTLA-4, only with PD-1. So those are exhausted cells, and these are the cells that John Wherry and Rafi Ahmed and others have concentrated on in their animal models, because these are cells after chronic antigen exposure approach this epigenetically defined state where they have PD-1, LAG3, and TIM3. Again, they're not going anywhere without help. They still have that phenotype, though, when you give the antibody. So what that tells us is if the antibody goes away, those cells are going to stop dividing. But the fix of that is temporary, and you haven't changed anything. So they're going to stop dividing as soon as you quit giving the antibody. And so you kind of expect